Hi, everyone. Good morning. My name is Alexis Chun, and this summer I've had the privilege of working in Dr. Maria Harrison's lab under the guidance of my mentor, Shiki Zhang, working on a project on optimizing a deep learning-based program that quantifies arbuscular mycorrhizal fungal colonization. So what is the arbuscular mycorrhizal symbiosis, or AM symbiosis? This is a symbiotic interaction between plants and fungi in which the plants provide carbon to the fungi in the form of sugars and lipids in exchange for essential nutrients such as phosphorus and nitrogen. And the fungi create a network in the soil, seen in this image, that expands far past the area that's taken up by the plant's roots. So these are the plant's roots right here, and as you can see, the network of the fungi is so much larger than that. And this provides access to nutrients in the soil that are otherwise unreachable by the plant. This is one of the oldest and most ubiquitous symbiotic interactions, having existed for hundreds of millions of years in over 70% of flowering plants. In addition to this, the interaction is also widely studied for its agricultural potential for increasing nutrient use efficiency. So how exactly do the plant and the fungi interact? Well, the fungi colonize the plant by penetrating through the plant's roots, through the epidermis and into the cortex as intraradical hyphae, which are seen by these purple lines here. And upon reaching the cortex, the hyphae branch in the cortical cells, creating these tree-like structures called arbuscules. And arbuscules are important because they're the key sites of nutrient exchange. And this is a picture of a mature arbuscule under a microscope. Other notable structures are the vesicles, which are seen here and in this image. And these provide storage for lipids for the fungi. Now, the fungi don't just colonize the roots at a single location. They are capable of simultaneously colonizing the roots at different points or at different times, resulting in many colonized regions along the plant root system of different levels of maturity. So this image is showing 3 different areas where colonization has occurred, resulting in what are called infection units. And these are made up of the structures that form from a single entry into the root. And this graphic over here is showing 3 different infection units as well that have been formed from 3 different entrances into the root, making up a greater colonized region. So why exactly is colonization important, and why do we care about measuring it? Well, colonization level is a really important parameter for characterizing genes and proteins that affect the AM symbiosis. For example, one protein that the Harrison lab has previously studied is the metacargo-trincatula phosphate transporter 4, or MTPT4. Our lab found that this was a really important protein because it affects the structures that are formed during colonization and colonization levels as a whole. So this image over here is showing arbuscules that have formed in a wild type plant. These arbuscules show the characteristic large tree-like highly branched structures, while these arbuscules were formed in a mutant that had a deletion of the gene that codes for the MTPT4 protein. And we can see much smaller degenerated arbuscules. This graph over here is also showing the difference in colonized root length as a whole across the whole root system. So the wild type is showing a much higher level of colonization than the MTPT4 mutant, again indicating the importance of this gene and protein for the successful symbiosis. So now that we know why colonization levels are important, how do we measure them? The most common method for measuring colonization levels is called the grid intersect method. This method involves randomly dispersing a set of roots onto a plate that's marked with a grid, as shown here, and viewing this plate under a microscope. The places where a root intersects with a grid line is counted, noting whether the root was colonized or non-colonized at that intersection. Once all of the grid lines have been viewed, the counter will be left with a number of intersections of colonized roots and a number of intersections of non-colonized roots. And these numbers can be used to give an approximate count of the colonization level across the whole root system using this calculation. So the total number of colonized intersections is divided by the total root intersections. Now, this is a really simplified view of what this process normally looks like. And this is an example of a set of roots that I was counting. And as you can see, it's a little bit more complicated. We have some places where roots overlap, and there can be instances where roots clump together, and it's difficult to get an accurate count of how many roots there are or to determine whether roots are colonized or non-colonized at those places. There are also places where roots might not fully intersect with a grid line, or where roots are colonized at one point of intersecting and non-colonized at another. So it's up to the counter to determine how to characterize those roots and to keep those characterizations consistent as they continue counting. Overall, this results in a method that's really time-consuming and also largely subjective and prone to human error. So given the flaws of the grid intersect method, a program was recently developed by a lab at the University of Cambridge called the Arbuscular Mycorrhizal Finder, or AmFinder. This is a deep learning-based program that uses image recognition software to determine whether roots are colonized or non-colonized in an image. The AmFinder uses two convolutional neural networks, or CNNs, which are mathematical algorithms that are used primarily for image recognition. The way that this program works is an image is inputted, and the program divides the image into tiles, as shown here. Then the tiles are either designated as containing colonized root sections, non-colonized root sections, or background. So the objective for my project this summer was to adapt AmFinder for use in our lab so that way we could more efficiently quantify fungal colonization levels in roots. To do this, I first needed to optimize the root staining procedure. So in order for colonization levels in roots to be visualized, the roots must first be stained. And there are a number of different methods to do so, but the program was originally trained on ink-stained roots, and so that was the method that we would be using. We also needed to determine the ideal image capturing method. Now, the paper that the program was originally published with describes two different methods of image capture, flatbed scanner and light microscope. So we plan to compare those two methods. And we finally needed to increase the accuracy of the AmFinder predictions, by creating a data set of our newly taken images and using them to train the program. The program does come pre-trained. However, we were unsure whether the training that was provided already would be sufficient for the images that we would be taking. So we plan to create our own data set of new images to retrain the program. So like I mentioned, the first thing that we did was optimize our ink staining method, and this method isn't really commonly used in the Harrison lab, so we did have some trial and error to work through. The first thing that we did was change our conditions for the KOH clearing step. This step is necessary because it clears the dark brown background color of the roots and allows for stained structures to be visible once the roots are stained. But if they're left in KOH for too long or at too high a temperature, the roots can become really brittle and hard to work with. So we determined that the best conditions for this step were to leave the roots in the water bath at 95 degrees C for 90 minutes so we could monitor the clearing process. We then needed to change the time that we were leaving our roots in the actual ink vinegar solution for, because we originally started out with 10 minutes, but even after destaining, this left us with roots that were far too dark for the structures to be clearly visible under the microscope even. So once we got it down to 5 minutes, we were left with much better results. Finally, we changed our destaining procedure. We were working originally according to some old procedures that our lab had that said that we could use DI water and a drop of vinegar to destain the roots, but this didn't really help us clear the excess staining solution very well. So we switched to this Clear2 solution, which is recommended by the paper that AmFinder is published with, and this left us with much better visibility of the stained structures. So now that we had a reliable staining procedure, the next thing we did was move on to our imaging method. We started off by using a dissecting microscope, but we quickly realized that this just wasn't a feasible method for imaging. The reason being is that even on the dissecting microscope, the field of view is just far too small to efficiently visualize our roots. We would have had to take at least dozens of images just to see one root system, and because we're ultimately trying to make this a more efficient process for our lab, this just wasn't the best course of action. So we moved on to the flatbed scanner, which is shown right here. And this isn't anything fancy. It's just a normal office paper scanner. But because it delivers such high resolution images, we were able to get some great visualization of our roots. So this is what the normal setup looks like with the roots in this red square over here. And after the scan is complete, we get an image that looks like this. And I know it just looks like a big dark blob of roots, but when we zoom in, we can see structures in the roots. So this root back here is an example of a non-colonized root, and then the roots up here in the front are colonized roots. And we can see some vesicles and some arbuscules going through the roots here. And most importantly, we can distinguish non-colonized roots from colonized roots, which is what we were aiming for. So now that we had obtained the images of our stained roots, we were able to use these images to train the program. The way that this is done is in the graphical interface of the program called AMF browser. This is a picture of AMF browser, and this is where we can annotate the images and tell the program which pieces of the image are colonized root segments and where are non-colonized root segments. So this is a picture where all of the annotations in AMF browser have already been filled out from a prediction that was run on the image. This red box here is showing us where this more zoomed in picture is looking at, and we can see that this piece of root has been accurately identified by the program as non-colonized. Now, annotations can also be filled out in the program manually by the user, and predictions that the program gives us can be edited by the user in AMF browser. So once all of the annotations have been filled out and when we know that they're correct, we can use these annotated images to compile a data set for the program to be trained with. The way that training works is that the program runs through all of the images, making its own predictions on the images, and it compares those predictions to the annotations that were provided by the user. Once a training cycle is complete, a new set of images has to be compiled, and predictions can be run on those images through the program, and the accuracy of the program can be reevaluated. So in order to calculate the accuracy, we use this equation here, where the true positive rate is the number of root intersections that have been accurately identified as colonized, and the true negative rate is the number of roots that have been accurately identified as non-colonized, over the total number of colonized roots plus the total number of non-colonized roots. Now, the original accuracy of the program that we calculated before any training had happened was about 69%. We were able to get the accuracy of our program after training to about 74%. And we think that with additional training with much larger data sets, we'll be able to get this accuracy to around 95%, because that's what the paper that the program is published in reports as the program being able to achieve. Furthermore, colonized roots can also be visualized with the fluorescent antibody staining, this WGA Alexis 488. And when these stained roots are visualized under the microscope, we see this really pretty bright green color where stained structures are. This is the staining procedure that's most commonly used by the Harrison lab, and since we're trying to get this program usable for our lab, it would be ideal if we could use this staining method in conjunction with the program. However, we ran into a couple of problems with this. So this is a picture of a set of roots under the normal bright field view of a microscope. But when we look at the roots with the fluorescent filter on, we can only see this small colonized area at the top. Obviously, this is a problem for AmFinder because it needs to be able to visualize both the colonized and non-colonized roots. We're working on a couple methods to fix this, including maybe trying to overlay the two pictures in an image processing software or something like that, but we still haven't done it. We are also left with the problem of the field of view of the microscope being far too small for efficient imaging. Now, we haven't been able to solve this problem yet, but maybe there are different fluorescent scanners that we can use that we still have to try. So in conclusion, AmFinder is functional. However, the accuracy is not where we need it to be in order for it to be functional for lab use. In order to get the program to 95% accuracy, at which point it will be able to use, we need to compile much larger data sets to further train the program. We are also not currently able to visualize WGA stained roots and quantify them with the program. However, given that an appropriate imaging method is found, we should be able to compile a data set of these images and get the accuracy on those images up to 95% as well. So I'd finally like to acknowledge all of the people in my lab, especially the people in the Harrison lab, Dr. Maria Harrison and my mentor, Shiki Zhang, and all of these other people and the people who funded this program and made this research possible.